In this discussion, we'll be looking at the anatomy of the male and female reproductive systems. Now, unlike the structure of some other topics that we've covered, where we've looked at the anatomy and then the physiology of a particular system, it's going to appear first as if we're focusing mostly on the anatomy here, when the reality is much of our reproductive physiology would be included under our endocrine discussion with our secretions of estrogen, FSH, LH, all driving processes within this reproductive anatomy. So as far as our reproductive systems specifically go, it's important to know the relevant anatomy. And because of that, we're mostly just going to be dealing with the diagrams and discussing some of the relevant structures and their functions as we go. Start with the female reproductive system, external anatomy first. You would have covered these terms in lab. Of course, we have two distinct folds of tissue, the labia minora and the labia majora, with the minora being more medial. As the labia minora comes to a point anteriorly, we will get the sensory organ of the clitoris, and just posterior to that, we will get to the urethral opening. One of the first things I want everybody to do is orient yourself with regard to the other systems being discussed. And we can most clearly get a true appreciation of that when we look at the lateral view. Here was our clitoris, anterior. Posterior to that, we had our urethral opening. Of course, that's going to be for micturition. So our bladder is going to be anterior to any of our reproductive organs, because our vaginal opening was posterior to the urethral opening, and our digestive organs will be posterior to it. For both the female and the male reproductive internal organs, I like to pick a starting point and work the anatomy from there. Again, I'm a big advocate of learning things in a sequence if we can, and it will help you fill in the gaps in your memory while you're looking at models in lab or diagrams as part of the lecture. With the female reproductive system, I always find it useful to start with the ovary, work your way into the uterus from there. We will look at the structure of the ovary specifically on our next slide, but we have a left and a right ovary that's going to be lateral to the main structure of our uterus. The ovary is where we're going to see maturation of the egg within the follicle. And upon ovulation, that mature oocyte is going to be projected by way of the fimbriae into the fallopian tube. The fallopian tube is going to be that internal lining that the oocyte is going to make as it makes its way to the uterus. Now, complete structure of that tube is going to be referred to as the uterine tube. So as you look at our diagram, the fallopian tube is contained inside the uterine tube. We make our way to the muscular organ of the uterus. We've got the main body of the uterus, roughly forming a V, and we have the fundus forming more of a domed top to that uterus. Now we are going to have three distinct tissue layers to that uterus. We're going to have an outer epithelial lining, lined with connective tissue on top of it. Just underneath it, we will have a myometrium, a thick muscular layer. Remember, this is very much a muscular organ. Remember our discussions of our positive feedback loop and our birthing process with the contraction of the uterine muscle. Internal to this, we have our endometrium. Now, this is going to be the lining that we'll refer to as being sloughed off as the female goes through its 28-day cycle. We will look at that cycle with regard to the reproductive hormonal control with our next topic. We start moving inferior, and we meet the cervix. We have our cervix aperture, the dome surface of the cervix inferiorly, and the invaginated portion is called the fornix around the opening of the cervix. We continue inferiorly and we reach the opening of the vagina. Now, I said we'd get to a diagram of the ovary, and here we are. It's important to realize that this is not an anatomical drawing of the ovary, but rather a functional drawing of the ovary. As you can see, it's numbered. So we're looking at the maturation of an oocyte, an egg, within the follicle, until at which point we get ovulation, a release of that oocyte from the ovary, and then we see functional tissue left behind, and then ultimately what the remnants of that follicle degenerates, and we will go around that cycle every 28 days. So we start off with what we refer to as our primordial follicles. These are going to be very immature follicles. Females are born with a certain number of follicles that then mature one at a time, along with the monthly cycle. We mature into a primary follicle, still very much immature, with an immature oocyte contained in the middle. That oocyte at that point is going to be surrounded by the zona pellucida. It's a thick, transparent membrane that surrounds the ovum, and that's still going to be even present later on upon ovulation. We have our granulosa cells surrounding that zona pellucida. Those are going to be cells that are going to respond to FSH for the conversion of androgens into estrogen. And then superficial to it, we get our thecal cells. Cells that are going to produce androgens in response to our luteinizing hormone. We move from our primary follicle into our secondary follicle, and a hallmark physical feature of it is going to be the beginnings of our antrum. Antrum is going to be a fluid-filled portion within the follicle at this point, and it's the first production of that antrum that's going to indicate the maturation into a secondary follicle. Time proceeds, we mature into a Graafian follicle. The atrium has now become very well developed. We have our cona radiata. It's going to be directly adjacent to our previously described zona pellucida, which is at this point in time the inner protective glycoprotein layer of that oocyte. The cona radiata is going to serve to supply vital proteins to the oocyte itself. Finally, we see our oocyte break free of our mature follicle, still surrounded with that zona radiata tissue. It's important to realize physiologically that upon ovulation, our discussion doesn't end there. We still have to look at the remnants of that follicle in which the mature oocyte left. Because while this oocyte may be heading down the fallopian tube now and headed towards the uterus to maybe get implanted and fertilized, the leftover corpus luteum now becomes very much endocrine tissue. The corpus luteum starts to produce progesterone, which helps maintain our uterine lining. If that oocyte that we just ovulated has any hope of being fertilized and implanted, we need a robust endometrial lining. So the oocyte leaves the ovary. The leftover corpus luteum produces progesterone that maintains that endometrial lining while the mature egg is making its way down to the uterus. If that egg does not implant into the endometrial lining at a certain period of time, then we will see our corpus luteum degenerate into a remnant corpus albicans. At that point, we're not producing any more progesterone anymore. Progesterone levels drop. We lose that maintenance of the endometrial lining. It sloughs off, and at that point, there is no potential for an embedding of a fertilized egg into the inside of the uterus. From there, we move even more superficial, looking at the structures that anchor the uterus, the ovaries, and the fallopian tube within that lower pelvic cavity. Generally speaking, we get a number of cord-like ligaments projecting from our reproductive anatomy. we get ovarian suspensory ligaments projecting superior and lateral to attach onto the inside structures of our pelvic cavity. We have our round ligament of the uterus. Eventually goes on to attach onto the inner tissue that's adjacent with our labia majora. And we get a bit of a thickening of a fold of tissue that's going to run adjacent to our left and right ureters. Lastly, we get another ovarian ligament, which will attach and tether the ovary along with our suspensory ligament onto the medial side of our outer ovarian wall. So we're kind of tethering the ovary in between those 2 points. Everything else is described as, at least in portion, to our broad ligament. We got our mesometrium of the broad ligament, which would be the larger portion. We got our mesosalpinx of our broad ligament, which will be just superior to our ovarian ligament. And lastly, we're left with our uterosacral ligament. It's going to tether the bottom of the uterus and project onto the sacrum internally. We started with the ovary and worked our way through the anatomy in the female reproductive system. In the male, we start with the testes. We have the testes. We have our sperm-producing structure inside the scrotum, made up of a combination of our seminiferous tubules, in which sperm is made, and our tunica albuginea, connective tissue meant to give the testes some form and some shape. Surrounding the testes, we have a slightly fluid-filled cavity called the tunica vaginalis, meant to reduce friction of the outer surface of the testes with the surrounding tissue. We will look at the seminiferous tubule here in just a little bit as far as sperm production. But ultimately, picture a tube with sperm being produced from the wall of that tube. Here's our seminiferous tube. Deep in the tube is where we're going to see our immature sperm, those primordial germ cells. Here would be the lumen of that tubule, the hollowed out inner portion. And generally speaking, we are going to watch the maturation of a sperm from the deeper portions furthest from the lumen towards the lumen of the tubule until finally we see mature sperm project from the inside wall of the seminiferous tubule and make its way through the anatomy. We see our primordial germ cell develop into our spermatogonium, which then matures into a spermacyte, undergoes meiotic division to half the genetic material. The primary spermacyte develops into a secondary spermacyte. We undergo meiosis one more time to produce our spermatids, until ultimately we produce our mature sperm. So our germ cell turning into spermatogonium, then a primary spermacyte, a secondary spermacyte, a spermatid, and then a mature sperm cell. So we see our sperm project from the inner lumen of those tubules. And then we look at everything after that as a continuous tubular duct in order to carry the sperm from the testes until it reaches the external environment upon ejaculation. We see the seminiferous tubules converge within the testes at a straight tubule. So we see the seminiferous tubules arranged in these lobule structures, which funnel into our straight tubule. Those are going to drain into our ret testis structures. Continuous flow, we go from the retesti into the epididymis. First, the head of the epididymis, still very much segmental. Ultimately, it's going to all converge into a single tubule within the body of the epididymis, through the duct of the epididymis, through the tail of the epididymis. Again, remember these in sequential order, right? Seminiferous tubule to our straight tubule, our retesti, our head of the epididymis, the body of the epididymis, the tail of the epididymis. Ultimately, we wind up in our vas deferens, which is going to be our tubule that becomes part of the spermatic cord. That vas deferens, can't see it on this diagram because it's going to be lateral, but picture it's going to run up lateral to our urinary bladder. And you can see it pick up again right here. So where the red line is, picture that being behind the anatomy. It's going to run over the top of our bladder, continue around the posterior aspect, where then it's going to meet our seminal vesicles. Now from there, we kind of have a bit of a step-by-step process to sperm production. And ultimately, we're going to wind up with converging anatomy with our urinary system. The seminal vesicles are glandular tissues, exocrine glandular tissues, that contribute approximately 85% of all of the fluid to the semen. We see the seminal vesicles merge with our vas deferens. Then we reach our prostate. Basically a walnut-sized gland that has an opening for our urethra leaving the bladder, but also the ejaculatory duct. That's going to be the duct work that's going to be formed as the seminal vesicle meets with our previously described vas deferens. So the vas deferens is continuous with this ejaculatory duct after the point the seminal vesicles contribute their component. We said before we weren't going to describe the portions of the male urethra until we got to reproductive. The portion of the urethra that passes through the prostate is logically called the prostatic urethra. And at the point where the ejaculatory duct drains into the prostatic urethra, from that point forward, we've got completely shared anatomy between reproductive, male reproductive, and urinary. We exit the prostate and we immediately enter our membranous portion of our urethra. While in this smallest portion, we are going to see a contribution to our seminal fluids from the bulbourethral gland, also called a Cowper's gland. We're going to see the bulbourethral gland continue to contribute a final volume to the semen, and it's going to produce a lubricating mucus secretion. Helps lubricate the semen as it exits the remainder of the urethra. Once we leave the bulbourethral gland, we're still in our urethra. Now we're very much part of the penis. And any sperm and the components that we had add to the semen by the seminal vesicles, the prostate gland, and the bulbourethral gland will now be ejaculated from the opening of the penis. So I encourage you to learn these structures from the diagrams. Very often, the location of the diagram, once you learn the function makes sense based on location. You should get accustomed to trailing these anatomical sequences from the starting point through to their finishing point, and that's going to help the memorization process. We finish up this topic by looking at hormonal control. And this is where the endocrine and the reproductive system really converges together. We'll talk about hormonal control of the reproductive system, and at the same time, we're going to add on 2 more layers to our hierarchy of hormonal control. But this time, looking at how the hypothalamus and the pituitary ultimately work to regulate spermatogenesis and our female reproductive system. And as usual, if you have any questions, please let me know. And we will be posting the final lecture next week.